How does recombinant DNA make monoclonal antibodies? Imagine being able to produce a perfectly tailored antibody in a lab instead of waiting for your immune system to do it naturally. That's what recombinant DNA technology makes possible. It's a process that allows scientists to create monoclonal antibodies with precision and consistency. These antibodies are powerful tools in fighting diseases and are used in many immunotherapies. The process begins with identifying the specific DNA sequences that code 4 parts of an antibody, especially the variable regions that determine what the antibody can recognize. These sequences can come from hybridoma cells, which are special cells that naturally produce monoclonal antibodies, or from antibody libraries created through display technologies. Once scientists find these sequences, they synthesize and copy them into circular DNA molecules called expression vectors. These vectors are like delivery trucks that carry the antibody genes into host cells. Next, the vectors are introduced into host cells, usually mammalian cells like Chinese hamster ovary cells or human embryonic kidney cells. These cells act like tiny factories, reading the genetic instructions in the vectors and making the antibodies heavy and light chains. Inside the cell, these chains come together to form a complete antibody. The cells then secrete these antibodies into the surrounding culture medium. Scientists collect the medium and purify the antibodies using special techniques called affinity chromatography. This process isolates the antibodies by binding them to specific molecules, making sure only the desired monoclonal antibodies are collected. Once purified, these antibodies are tested for their ability to bind to their target antigen, their strength, and how well they work. This whole process is a way to mimic and control natural antibody production, but in a laboratory setting. Instead of relying on animals or humans to produce antibodies, scientists use genetic instructions to produce consistent and customizable antibodies. They can improve the stability of these antibodies, reduce unwanted immune reactions, or change their species' origin to make them safer for therapy. In the world of immunotherapy, recombinant monoclonal antibodies are especially useful because they can be designed to target specific disease markers. For example, they can block cancer cell receptors or calm down autoimmune responses. This targeted approach helps the immune system intervene more effectively. Thanks to recombinant DNA technology, we can produce these specialized antibodies efficiently, making them an essential part of modern medicine.